Hello friends, my dear students, myself Wandhare Sheetal from XII.SCI's Rao Bahadur Narayan Rao Boroke College, Sirampur. Now from this college, the various videos on the theory chapter we have prepared, and these videos are already uploaded on the college website. These videos are prepared under the guidance and support of Honorable Principal Dr. Gaikwad Sahib. Now in this video, whatever the skeleton which is having get evolved, or we can say that modified by the or according to new syllabus of the state board, the practical skeleton question paper that we are going to discuss in this video. As we know that here nowadays the various online lectures program they are running because of the effect of COVID-19, and the various part of the syllabus is already get reduced by the HSC board. That is, as we already know, the 25% reduction takes place from the theory part, and same thing it will be get copied with the help of the practical also. That is, some practicals that will be get reduced, and whatever the content they have kept for the analysis for your regular practical examination, these things they will be get discussed on this video. Now your total practical is having the 30 marks, and this board is covered that all the pattern of the skeleton question paper. This total practical of the 13 marks that will be carries divided into the 3 parts, A, B and C, and each part is having the 10 marks weightage. There is a no option for the practical. Be remember that each and every student that these all the practicals they are compulsory to attend and compulsory to do when we are performing that at the time of the exam in the laboratory. The practical handbook is already available on the market. They will be also gives you the guideline how to get this demonstration, how to get experiment in the laboratory also. That same thing here we are discussing. Now let's start from the part A. This part A carries the major experiment as well as the minor experiment. This major experiment is having the 4 mark and the minor experiment is having the 3 marks. In this question number first, thus you have to perform the experiment with the help of the dissection box, the structure distribution of the stomata. And very firstly you should be clear that no practical is out of the syllabus. Whatever the things you are studying in the theory chapters, that is each practical is based on the chapter. Means if you are clear in your theory, at that time you will be get very clear in practical also, because these practicals they are having some background, some theories related to that, and on that basis the dissection, slide preparation, these things you have to perform in the laboratory. Now in this section first, the stomata, it is the very simply, that is nothing but the respiratory part of the plant, or we can say that here the 90% the process of the transpiration takes place with the help of the stomata. The upper and lower leaf that carries the distribution of the stomata, as we know that here in the theoretical part, the maximum number of the stomata located on the lower surface because of to avoid the transpiration. Plant has to provide the content of the water, and for that purpose they will be shows that the maximum number of the stomata they are located on the lower surface. And what you have to do in this practical, you have to collect these leaves which will be get provided at the time of the practical with the help of the dissection box, taking the peel off, prepare the slide and observe this slide under the microscope, show that preparation to the examiner. Now this also showing the dividation, how to prepare the slide, how to identify that slide and show this slide to the examiner, and sketch and label that structure of the stomata. These things you have to perform in that practical, the question number first. For that also there is having the alternative, that is the adulteration of the milk. In these processes here, the effect of the starch, how it will be get takes place on the milk adulteration, that you have to get study, whether it will be get in the pure form, whether it will be get in the impure form. These things we are going to discuss in this adulteration of milk. Now in the minor experiment, the dissection, same thing takes place. You have to get the flower at the time of the practical. This floral parts you have to get separate. The floral part, that is nothing but the very simple and basic concept in the science student, because the identification of this floral part, how the location of the ovary is there, how you are going to take the TS of the ovary, you have to get observe these floral parts with the help of the naked eye, and the structure of the ovary with the help of the microscope. Here you have to use only the simple microscope for the identification of the ovary. The TS of the ovary that you have to take, this experiment also carries here the 3 marks. Now alternative for that question, that is nothing but the quadrat method. In this quadrat method, the which species is the dominant in the respected region or the area which have to get find out. With the help of this quadrat, the dominant species you have to get identify. With the help of the formula, the calculation you have to done at the time of the practical examination, prepare a chart. This chart is also shown in the practical handbook also, and the 1, 2, 3 whatever species are observing in a single quadrant, identify these species, or if you are unable to identify that such species, maybe you can write species A, species B, species C, and you have to find out the frequency of these species which is present in this quadrat method. Okay. Now in question number 3, this practical is also having the correlation with the help of the environment, because various chapters you have studied on the basis of the environment, and the find out the clarity and pH of the water sample, find out the soil pH and soil type of the water sample. These from 1 to 3 they are very compulsory practicals to all of you. In this here you have to supplied with the 2 different types of the water samples which are collected from the different water bodies which is present near your college areas, which is present near your villages also. Maybe there are various alternatives for that. Maybe this sample is having the acidic, neutral, basic pH, that you have to identify with the help of the pH paper. Whether this collected water, it is the clear water or unclear water, that is also you have to get identified. Then you have to find the soil pH. Again same thing you have to repeat, whether this soil is having the acidic nature, whether this soil is having the alkaline nature, basic nature, maybe it can be used for the agriculture purpose or not can be used for the agriculture purpose. That things you have to get conclude at the time of the practical examination when you are preparing an observation table for these soil samples provided to you. Same conclusion you have to write related to the water bodies also, because whether these water having the acidic property, these water having the alkaline property, they are never used for the drinking purposes, agriculture purposes, that thing you have to mention in your conclusion. Okay, in this way here these 3 practicals are there. Now come towards the part B of this practical. This part B, it is showing the spots from A to the J, and again these spots they are from your theory. Whatever the theory chapters you have studied, these theory chapters, on the basis of that these practicals they are get based. Now as same pattern is running on the samely whatever the exam you have given in the 11th standard, that is each spot is carries the 1 mark, means this part B is again for the 10 marks. This total 10 spots included by the board from this new year and from this new syllabus, and the very previously they are having the less weightage for the spotting, but now it is having the maximum weightage for the spotting, that is about the 10 marks. This part showing or the part B showing the 10 marks. Let's see one by one. Here the spot A, this spot A, it is showing the adaptation. You should know here the concept of the pollination also. You have studied already in this the morphology of the flowering plant in case of the reproduction, in case of the angiosperm, how the angiospermic plant behave, how the fertilization takes place, how the pollination takes place, and the types of the pollination on the basis of the wind, on the basis of the insect, that you have to identify. In case of the spotting any XYZ type of the flower, or suppose the salvia flower is kept in front of you, then we have to identify that this flower, it takes the pollination by which way. If the maize flower is kept in front of you, the pollination it will be takes place by which way. That is important. This may be the insect pollination or that may be the wind pollination. That identification carries the half mark, and the remaining half mark is for the description. Now, alternative for that spot A, the stages of the hybridization, the emasculation, the removal of the anther, then the bagging as well as the tagging. These stages of the identification, they have been kept in front of you, and you have to identify these stages and describe them. Then in the spot B, the spot B, it is the demonstration experiment. In this demonstration experiment here, they are showing the 4-leaf experiment. That 4-leaf experiment, it maybe shows the transpiration. This transpiration, you have to identify with the help of this, the given experiment is of dash type, and whatever the changes takes place within this 4-leaf, that you have to describe. Again, alternative for that, the imbibition of the seed as well as the resin. The identify that whether seed that will be get imbibed, how this imbibition process takes place, that you have to explain. Again, the alternative for that in this spot B, the separation of the pigment by the paper chromatography. The different pigments, they will be get separated one by one. The very firstly, the chlorophyll it is get separated. After that, the carotene, xanthophyll, these pigments they are separated, and which range they are separated, that you have to identify, and that such a description it is accepted in your answer sheet. In the spot C, here that spot C, it is based on the adaptation. This adaptation, it is related to the plants, whether they may be the aquatic plant, they may be the xerophytic plant. This aquatic and xerophytic plant you have studied in your theory. Out of this, here any one plant is kept in front of you, and that shows that you have to identify this plant. Suppose here the Opuntia is kept in front of you, it is here the xerophytic plant. Suppose the Hydrilla is kept in front of you, Ecornia is kept in front of you, that is they are all the aquatic plants. Their identification is necessary, which are the characters they are performed due to for the adaptation purpose, or how these plants they can be adapted in that such a condition, that you have to explain with the help of mentioning their characters. Then the D spot, this is based on here the chapter that is the reproduction in lower and higher animals. Here the TS of the testis, TS of the ovary as well as the VS of the blastula. This ovary, it belongs to the female reproductive system. Testis, they are belongs to the male reproductive system. This VS of the blastula, it is nothing but the after fertilization, whatever the stages of the development that takes place, these stages of development that will be get explained here, and these slides, these permanent slides are observed under the microscope. After observation, describe these slides, or whatever the content you have observed, whatever the cells you have observed in this slide, that you have to describe very correctly. Then the pedigree chart. This pedigree chart, they are indicating whatever the genetic traits change takes place, that is get explained. Diseases and control measure. Means here various types of the pathogens they have been kept, not the diseases they have been kept in front of you. Just like that, suppose the typhoid, the causative organism for the typhoid, Salmonella typhi. The malaria, causative organism for that, the Plasmodium. Means the various images of the pathogen that is kept there, and that images you have to identify. Maybe here that will be shows that the indication this such a type of the pathogen is responsible to cause that such type of the diseases. Suppose the typhoid it is showing, or the Salmonella typhi image, it will be shows that the causative organism of the typhoid. You have to identify the disease and you have to describe only the symptoms along with the control measure. Now the stages of the meiosis. These stages of the meiosis you have already studied in the 11th standard. Now from the meiosis first, meiosis second, various stages are there. This meiosis first again divided into the various stages. Then the meiosis second, that will be follows the here prophase, metaphase, anaphase, telophase. That stages you have to identify and describe. Then in the spot H, this H spot here samely shows the adaptation repeated related to. Instead of the plants here, that will be get replaced by the animals. Maybe suppose the aquatic animals are common, that is nothing but the fishes, right? And the zero pole animals, that is the camel. These images they are here, and you have to identify these images and describe these images. Now the spot I, this spot I, it is showing the VS of the ovule. The various parts of this ovule that will be shown, you have to describe the functions of these parts which are shown on that structure. Then the pedigree chart. This pedigree chart may be here the color blindness, maybe the rolling of the tongue, or they are showing the various genetical trait changes takes place from parents to offspring, and how these changes takes place, that thing you have to describe by observing this pedigree chart. Don't rewrite that pedigree chart in your answer. Only observe this pedigree chart and write this pedigree chart's character. Suppose the color blindness is there. This color blindness is what? That is nothing but the person is unable to distinguish between the color. That is get explained here. Okay. Now the X chromosomal disorder, that is showing the X chromosomal disorder, and these things that will be explained here. Okay. Now the part C. This part C, it again carries the 10 mark. Here the question number 5th, it's showing the project report. The project you have given on the basis of the theory chapter, and this shows that you have to complete this project by following the scientific step. That is very firstly the aim of the project, that is the aim and objective you have to fix. After that, write the introduction. After that, whatever the references you have collected, whatever the methodology you are using for that project work, and the result, conclusion, what is get the result and conclusion of this project, and what is the need for that project to the society, or how this project is used to the society. Then the Viva. This question number 6 is showing the Viva, which is based on the practicals you have done on the basis of the part A, or whatever the syllabus you have studied from the 12th standard. On that basis, the examiner they are going to ask the question. Then the question number 7, it shows that the certified journal. This certified journal is for the 3 marks. In this way here, this total practical, it covers the 13 mark, and hope so that this skeleton, it will be gives you the very important guideline how to write the journal or how to study during this 12th science practical. That is showing here the skeleton of the question paper. Thank you.